Good morning, students. Today we shall start the second topic of our chapter, that is the genetics and evolution, that is the molecular basis of genetics. Before that, the heredity and variation we have studied. I hope that you understood it properly, because when you understood that chapter, then only it would be easy to understand the molecular basis of genetics. Okay. So, as the name denotes itself, molecular basis of genetics. Before that, whatever we have studied in the Mendel's law and the other gene interaction, we only talked about the gene or the factor, but we could not understand that actually what is this molecule, actually what is the gene and how it function, how it moves from one generation to another. अगर we say that genes move from one generation to another generation, in what way actually body से क्या move on होता है? Okay, so what is it? We are, as we have said, everything is in our body, nothing but the chemicals. तो ये किस तरह के chemicals हैं और what is actually the genetic material, right? We have said that gene is nothing but a piece of DNA. तो हमें पूरा जो पढ़ना है, वी हैव टू टॉक अबाउट द डीएनए, देन आरएनए और फिर प्रोटीन सिंथेसिस, बिकॉज जीन एक्सप्रेस देमसेल्व्स इन द फॉर्म ऑफ द प्रोटीन सिंथेसिस. तो इफ वी आर सेइंग जीन मीन्स इट्स अ पीस ऑफ डीएनए, राइट? So DNA is a main place which code the information कि किस तरह का expression characters का होगा किसी organisms में. As well as the RNA is important, because RNA plays as a messenger or the adapter, okay, so that the messages can be taken out of the nucleus and afterward the protein synthesis. So all these things we shall study here. First of all, that as we have said, DNA is a genetic material. तो कैसे उन्हें पता पड़ा साइंटिस्ट को कि DNA ही genetic material है? Because first of all, the protein is considered as a genetic material. Protein was considered as a genetic material because the protein is very big molecule and it can store lots of information. तो पहले प्रोटीन को कंसिडर किया गया कि मे बी इट इज अ जेनेटिक मटेरियल होगा ये, बट आफ्टरवर्ड scientists came to know and they have said that actually the DNA is a genetic material. Anything which is act as a genetic material should have certain characteristic features. पहला फीचर क्या होना चाहिए? That it should be a large molecule. जो भी जेनेटिक मटेरियल होगा वो कैसा होना चाहिए? इट शुड बी अ लार्ज मॉलिक्यूल, वेरी वेरी लार्ज मॉलिक्यूल. सो डीएनए इज वेरी लार्ज मॉलिक्यूल. दूसरा, इट शुड बी स्टेबल. इट शुड बी स्टेबल बोथ केमिकली एज वेल एज स्ट्रक्चरली. स्ट्रक्चर वाइज or chemical wise. Structure means whatever the arrangements of the different atoms are there, it should not be changed, और उसका chemical जो composition है वो भी change नहीं होना चाहिए. So structurally and chemically it should be stable compound. Isn't it कि बहुत ज़रा सा temperature change और pressure change होने पे the molecule should not change. So it should be stable. So DNA is a very stable molecule. Third is, it should be generate its replica. It should be duplicate. Replication also known as the duplication. Okay, अपने same copy बनाने की process को बोलते हैं replication. So the whatever genetic material we are considering, that molecule should have this capacity to generate its replica. अपनी copy बना सकता हो. Okay, so क्या है? It should generate its replica. Okay. Fourth point, that it should have the capacity of the mutation. Mutant. ये molecule ऐसा होना चाहिए जो जिसमें mutation हो सके. It can get mutated. So that only the evolution has been taken place, है ना, because of the DNA mutation. So it can mutate. So anything, whatever we are considering as a genetic material, should have these features. It should be a large molecule. It should be stable. It should generate its replica, and it should be mutant, a mutant molecule. It can get mutant. So all these features are found in the DNA, and then scientists considered the DNA as a genetic material, right? For this, the Frederick Griffith had done one experiment. Griffith, इसको Griffith experiment भी बोलते हैं. This is the Griffith's experiment. Frederick Griffith did one experiment. He was working on the bacteria Streptococcus pneumoniae for the bacteria who cause which is a pathogen for the pneumonia. So he was working on the pneumonia. So what he did, that he cultured two kind of the bacteria. One bacteria he gave the name R strain or the rough strain. Rough strain bacteria. These are the Streptococcus pneumoniae bacteria. This bacteria cause pneumonia in the organisms. अगर ये bacteria किसी organism में inject कर दी जाए तो they cause the disease pneumonia. Okay. So Griffith did one experiment. He cultured two strain of bacteria. One is the rough strain. उनका जो outer surface थी वो उनकी colonies जो थी वो rough type की थी. And these are the harmless bacteria. Means they don't cause the pneumonia. Okay. And another strain is S strain or the smooth strain. And these are the harmful. These are the harmful bacteria. The bacteria those who cause, अगर ये किसी में inject कर दी जाए तो they cause the pneumonia. Okay. Then he did the experiment. What was the experiment? When he injected this R strain bacteria in the mice. Okay. Mice में inject किए तो first experiment क्या था? R strain bacteria is injected in mice. यहाँ पे आप लोग लिखोगे injected in mice, and he saw all the mice were alive. They did not die. They were alive. Because the R strain is harmless, as we have said, ये harmless bacteria है. और जब ये हम mice में चूहों में उनको inject किया तो सारे चूहे जो थे वो alive थे, वो नहीं मरे थे और alive थे. Okay, what was the second experiment? When S strain बैक्टीरिया, आप यहाँ लिखोगे इंजेक्टेड, जैसे मैंने यहाँ लिखा इंजेक्टेड इन द माइस, द माइस डाइड. ऑल द माइस डाइड. वो सारे जो चूहे थे वो मर गए. ओके. अब थर्ड एक्सपेरिमेंट व्हाट ही डिड, दैट ही हीट किल्ड द एस स्ट्रेन. हीट किल्ड एस स्ट्रेन. एस स्ट्रेन को ऐसे थोड़ा सा उन्होंने बैक्टीरिया को हल्का सा टेस्ट ट्यूब में गर्म कर लिया तो उसे बोलते हैं हीट किल्ड एस स्ट्रेन. एंड देन दीज हीट किल्ड एस स्ट्रेन बैक्टीरिया इंजेक्टेड इन द माइस. एंड व्हाट वाज द रिजल्ट, दैट माइस डिड नॉट डाई, दे अलाइव. वो भी अलाइव थे और मरे नहीं थे. ठीक है. फिर फोर्थ एक्सपेरिमेंट में क्या किया, ही मिक्स्ड द आर स्ट्रेन and the heat killed S strain bacteria. What he did in the fourth experiment, that he mixed R strain plus heat killed S strain. I am writing here. Fourth experiment में R strain plus heat killed S strain. And what was the result? होना क्या चाहिए था? That all the mice should have been alive, क्योंकि R strain harmless है और S strain harmful था, but when उसको heat kill किया गया, it converted into the harmless. तो सारे mice alive होने चाहिए थे, but for the it's a contrary to it, all the mice were died. They all were died. Then the Griffith reached on this conclusion that on the heat killing, जब S strain को kill कर दिया था, at that time certain molecules or the genetic material which is very stable, on the heating also that material was not destroyed, and when this material is mixed with the R strain, it converted R strain into the harmful genetic material, and then that is the reason that all the mice were died. Understood? Because when it was seen, उन्होंने और एक्सपेरिमेंट्स किए, जैसे जब उन्होंने इस एस जो किल थे उसके ऊपर एक्सपेरिमेंट किए, तो उन्होंने देखा कि जब प्रोटीन डाइजेस्टिव एंजाइम पहले यूज़ किया, तो जो एस स्ट्रेन का जितना भी प्रोटीन था वो डाइजेस्ट हो गया, लेकिन फिर भी उनको सेम रिजल्ट, तो प्रोटीन नहीं है इसका मतलब उसके लिए रिस्पॉन्सिबल। लेकिन जब उन्होंने डीएनए मतलब डीएनए डाइजेस्टिव एंजाइम यूज़ किए और डीएनए डाइजेस्ट हो गया, तब उनको ये वाला रिजल्ट नहीं मिला। तो what does it mean? That DNA is a genetic material. I hope you understand, and this experiment is very important. अगर if it is not given in the NCERT book, but still you have to do that. It is mentioned in your syllabus. What was the experiment which proved that DNA is a genetic material? I hope you understood कि यहाँ पर RNA जो हार्मलेस स्ट्रेन था उसको हार्मफुल किसने बनाया? कि एस हीट किल्ड एस स्ट्रेन था, हीट किलिंग से उसके ऑल द अदर कंपोनेंट्स दे डिस्ट्रॉयड, बट सिंस डीएनए इज अ वेरी स्टेबल मॉलिक्यूल, केमिकली ऑल्सो स्टेबल, तो हीट उसको डिस्ट्रॉय नहीं कर पाई थी उस डीएनए को, एंड दिस डीएनए कन्वर्टेड द आर स्ट्रेन डीएनए इनटू द harmful strain, and because of that the mice died. Understood? So this is, by this experiment it was proved that DNA is a genetic material, right? RNA also, although in certain organisms RNA also genetic material, जैसे वायरसेस में है, एड्स का वायरस है या फिर टोबैको मोज़ेक वायरस है, उसमें भी वायरसेस, जो वायरस में RNA जेनेटिक मटेरियल है, पर उसमें पहले रिवर्स ट्रांसक्रिप्शन होता है RNA से DNA, that is known as the reverse transcription, and then the further process is going on. लेकिन उसमें जेनेटिक मटेरियल RNA है, ओके, RNA is also very essential in all these genetical process or the expression of gene, but RNA act as a messenger and adapter, and the main function or the, we can say the main genetic material is the DNA, right? In next lecture we between RNA and DNA also we can see that the DNA is, between these two, DNA is more stable, and that is why RNA or DNA may be, our DNA is considered as a genetic material, because we are knowing that DNA is made up of the deoxyribose sugar which has only one OH group. Last year I taught you the biomolecules, उसमें आपको DNA RNA का structure पढ़ाया था, और DNA में it has only one OH group. On the other hand RNA has two OH group. RNA में two OH group होते हैं RNA की राइबोस शुगर में, and in the deoxyribose sugar there is only one OH group. So अगर ज़्यादा more the OH group, the molecule is more reactive, और अगर ज़्यादा reactive होगा इसका मतलब ये less stable है. Okay, so between RNA and DNA, why DNA is considered as a genetic material? Because it is more stable than RNA. Why? Because RNA has, in the ribose sugar, there are two OH group. More the OH group, the molecule is more reactive, मोर रिएक्टिव मीन्स इट इज लेस स्टेबल. स्टेबल नहीं होगा, वो रिएक्ट करने की कोशिश करता रहेगा. ऑन द अदर हैंड डीएनए हैज इन द डीऑक्सीराइबोज शुगर ओनली वन OH ग्रुप. तो दोनों में एक OH के साथ वो ज़्यादा स्टेबल, कम रिएक्टिव है और ज़्यादा स्टेबल है. On the other hand, we are knowing that in the RNA जो नाइट्रोजनस बेसिस होते हैं, एडेनाइन, ग्वानिन, साइटोसिन और थायमिन की जगह पे यूरेसिल होता है. RNA has the यूरेसिल नाइट्रोजनस बेस और द डीएनए हैज थायमिन. ओके, एडेनाइन, ग्वानिन, साइटोसिन तो होंगे ही, पर उसके साथ यूरेसिल होगा आरएनए में, विच इज अगेन मोर रिएक्टिव, और थायमिन इज लेस रिएक्टिव. थायमिन इज मोर स्टेबल. लेस रिएक्टिव का मतलब है मोर स्टेबल, एंड थायमिन इज फाउंड इन डीएनए. So again, because of this reason also, because uracil is more reactive, तो वो less stable है, और thiamine is more stable. So that is why again the DNA is proved as a more stable और ज़्यादा stable है. That is why it is appropriate for to being a genetic material. So all these are